An ancient killer, a survivor of mass extinctions, and a tormentor of shellfish has been unmasked. For nearly 500 million years, a parasitic worm has been waging a silent war, etching its distinctive mark into the shells of marine creatures. This isn't science fiction. It's a stunning revelation from the world of paleobiology. In the heart of Morocco, within rocks that whisper tales of a long-lost ocean, scientists have uncovered the handiwork of a 480-million-year-old worm, a creature whose parasitic legacy endures to this day, plaguing modern oyster beds and challenging our understanding of evolution itself. This is the story of a worm that cheated time, a master of survival whose story is written in stone. To understand the world of this ancient worm, we have to journey back in time to the Ordovician period, which spanned from about 485 to 443 million years ago. This was a time when the Earth was a very different place. The continents as we know them didn't exist. Instead, most land was gathered in the southern hemisphere, forming the supercontinent of Gondwana. The climate was an intense hothouse, with sea surface temperatures that may have soared to a blistering 45 degrees Celsius at the equator. Atmospheric carbon dioxide levels were a staggering 14 to 16 times higher than they are today, driven by widespread volcanic activity. This intense greenhouse effect led to some of the highest sea levels of the entire Paleozoic era, flooding the low-lying continents and creating vast, shallow inland seas. These warm, sunlit waters would become the cradle for an unprecedented explosion of life. While the preceding Cambrian period is famous for its Cambrian explosion, where most major animal body plans first appeared, the Ordovician witnessed its own remarkable evolutionary event, the Great Ordovician Biodiversification Event, or GOBI. Over about 25 million years, the number of marine genera quadrupled, fundamentally reshaping marine ecosystems. The seafloor, once dominated by creatures like trilobites, became a multi-tiered world of suspension feeders. Bryozoans, or moss animals, formed delicate branching colonies. The first true coral reefs began to grow. Brachiopods, with their two-part shells, became incredibly abundant, their shells carpeting the ocean floor. Echinoderms, the group including starfish and sea lilies, reached their peak diversity. Above the seafloor, new predators emerged. Cephalopods, the ancestors of modern squid and octopus, diversified and became dominant hunters. This was a world of increasing complexity, with the rise of mobility, predation, and, as our story reveals, parasitism. The intricate food webs being woven in these ancient seas set the stage for the next 200 million years of life. Our story's main setting is a remarkable fossil site in southeastern Morocco, known as the Fezouata Formation. Located near the town of Zagora on the edge of the Sahara Desert, this geological formation is a Lagerstätte, a German term for a deposit that exhibits extraordinary fossil preservation. What makes the Fezouata Formation so special is that it preserves not just the hard shells and bones of ancient creatures, but also their soft tissues. This is incredibly rare. The exceptional preservation gives paleontologists an unparalleled glimpse into the full diversity of life in the Ordovician seas, including the soft-bodied organisms that are usually lost to time. The Fezouata biota is a fascinating mix of old and new. It contains holdover species from the Cambrian period, creatures thought to have gone extinct long before, like the iconic Anomalocaridids, large swimming predators with grasping appendages. Alongside these Cambrian survivors are the earliest known examples of many groups that would dominate the Paleozoic and even modern oceans, like the first horseshoe crabs. The environment was a shallow sea on the continental shelf of Gondwana, which was then located over the South Pole. The fossils were likely buried rapidly by storm-stirred sediments, which is why they are so well preserved. It is within this remarkable fossil assemblage, among the shells of some of the earliest bivalves, that the evidence of our ancient parasite was found. The discovery was not the result of finding the worm itself. Soft-bodied creatures rarely fossilize. Instead, paleontologists rely on trace fossils, the marks and burrows these animals leave behind. In this case, the evidence was a series of peculiar, repeating patterns etched into the fossilized shells of early bivalves. A team of researchers, led by paleobiologist Karma Nanglu, noticed that the shells were peppered with small, distinctive markings. Nanglu explained, "The marks weren't random scratches. We saw 7 or 8 of these perfect question mark shapes on each shell fossil. That's a pattern." Initially, the team was mystified. The shape was so precise and consistent, it demanded an explanation. Another researcher, Javier Ortega Hernandez, recalled, "It took us a while to figure out the mystery behind these peculiar-looking traces. It was as if they were taunting us with their question mark-like shape." The answer, as is often the case in science, came from connecting these ancient traces to the work of a modern-day parasite. The researchers concluded that the question mark-shaped burrows were the work of a type of marine bristleworm belonging to a group called Spionids. This was their eureka moment. Modern spionid worms, particularly those from the genus Polydora, are well-known parasites of modern oysters and mussels. They bore into the shells of these bivalves, creating tunnels that can weaken the shell and even kill the host. The shape of the burrows made by modern spionids was a perfect match for the 480-million-year-old traces. To confirm their findings, the researchers turned to microcomputed tomography, or micro-CT scanning. Similar to a medical CT scan, this technique uses X-rays to create a detailed 3D virtual model of an object, including its internal structures. This non-destructive technique is a game-changer for paleontology, allowing scientists to peer inside rare fossils without destroying them. The micro-CT scans of the Fezoata bivalve shells revealed the question mark-shaped burrows in stunning 3-dimensional detail. They showed how the burrows penetrated the shell and even revealed previously unseen burrows hidden within the layers of rock. The 3D models confirmed that the shape was not a superficial scratch, but a complex structure created by a burrowing organism. This discovery provides a remarkable window into the life of a parasite that has been using the same strategy for nearly 500 million years. The culprits, both ancient and modern, are spionid worms, a family of polychaetes or bristleworms. The modern-day counterpart to our ancient worm, Polydora websteri, often called the oyster mudworm, provides a living model for understanding this ancient relationship. The life cycle begins with a larval stage that swims freely in the water. When a larva is ready, it seeks out a suitable hard surface, with a preference for the calcium carbonate shells of bivalves like oysters. Once settled, the young worm begins to bore. It does this through a combination of chemical and mechanical action. The worm produces an acid that dissolves the shell, and it uses its bristles and body movements to help shape the burrow. This is how the distinctive question mark shape is formed. The larva first attaches and dissolves a small area to anchor itself. This becomes the dot of the question mark. As the worm grows, it tunnels deeper into the shell, creating the curved shaft. This unique and complex burrow shape is a diagnostic feature. No other known organism creates this exact pattern. Inside its burrow, the worm is protected from predators. It extends 2 long tentacles, called palps, out of the burrow's opening to feed on detritus and plankton from the surrounding water. The Spionid worm is not a direct predator of the oyster. It doesn't eat its flesh. Instead, it's a kleptoparasite. It steals the oyster's food. By positioning its feeding tentacles near the oyster's own feeding currents, the worm intercepts food particles before the host can consume them. While not directly feeding on the host, the worm's activities are far from harmless. The burrowing process weakens the shell's structure, making the oyster more vulnerable. In response to the irritation, the oyster secretes a layer of nacre, the same material that forms pearls, to wall off the intruder. This creates a mud blister on the inside of the shell, a pocket filled with mud, feces, and the worm itself. In cases of heavy infestation, these blisters can interfere with the oyster's ability to close its shell properly and even damage its adductor muscle, which can be fatal. The most profound implication of this discovery is the incredible longevity of this parasitic relationship. The fact that Spionid worms were using the exact same method 480 million years ago is a stunning example of evolutionary stasis. As researcher Karma Nanglu put it, this group of worms hasn't changed its lifestyle in nearly 500 million years. We tend to think of evolution as constant change, but here's an example of a behavior that worked so well. It stayed the same through multiple mass extinction events. This parasitic strategy successfully weathered some of the most catastrophic events in Earth's history. The Ordovician's Silurian extinction, which wiped out an estimated 85% of marine species, didn't stop it. Neither did the devastating Permian-Triassic extinction, the Great Dying, which eliminated over 90% of all species. This humble worm, with its specialized lifestyle, has proven to be one of the most successful and resilient parasites in our planet's history. The long-term success of this relationship also offers a glimpse into the co-evolutionary arms race between parasites and their hosts. The formation of mud blisters by oysters is a clear defensive response. This suggests a long history of adaptation and counter-adaptation. However, the fact that the worms continue to thrive indicates that this conflict has reached a kind of stalemate, an ecological standoff that has persisted for eons. The study of such ancient parasite-host interactions is a growing field known as paleoparasitology. It's a challenging field, as parasites rarely leave direct fossil evidence. Discoveries like the Fezoata Spionid traces are therefore incredibly valuable, providing concrete proof of parasitic behavior in deep time. This story isn't just a tale of the deep past. It has very real implications today. The descendants of this ancient parasite are a significant problem for the modern aquaculture industry. Oyster farming is a multi-billion-dollar global enterprise, and infestations by shell-boring spionid worms like Polydora websteri can cause major economic losses. The mud blisters make oysters unsightly and reduce their market value, particularly for oysters served on the half-shell. Heavy infestations lead to decreased growth rates, poor meat quality, and increased mortality in oyster populations. The problem is exacerbated by global trade, which has spread these highly adaptable worms around the world. Aquaculture farmers are actively working on ways to mitigate the impact of these pests, using strategies like careful site selection and off-bottom culture techniques. The threat is also being amplified by climate change. Rising sea temperatures and ocean acidification can create a more favorable environment for the parasites while simultaneously stressing their oyster hosts, potentially tipping the long-standing balance of their co-evolutionary arms race in the worm's favor. Understanding the deep history of this relationship can provide crucial insights for predicting how it might respond to the changes we are causing today. The discovery of the 480 million-year-old Spionid burrows is a profound reminder of the interconnectedness of life through time. It's a story that links a remote fossil site in the Moroccan desert to the bustling oyster farms of today, and a humble bristle worm to the grand narrative of evolution. This ancient worm, with its persistent question-mark-shaped signature, is a symbol of resilience, a master of a parasitic craft perfected over hundreds of millions of years. Thanks to the keen eyes of paleontologists and the power of modern technology, the secrets of this ancient parasite have been unlocked from their stone tomb. It's a powerful illustration of how studying the past can illuminate the present and help us prepare for the future, a story that will continue to be told, not just in scientific journals, but in every oyster that bears the mark of this immortal killer.